Could you check it like after a few minutes?
Yeah, sure. Absolutely.
I'm gonna ask her to check it.
Okay.
I already started.
Okay.
Check it out.
Okay. Sure. Yeah, I'll sit here.
We had a lot of ice on you. We had driving.
Oh, yeah, everyone needs to drive. Oh, same thing in Alabama. Yeah. I don't have trucks, all
The big danger we had there
Or downtown.
was the flying fight, because
I
he's
see
like,
he's coming from.
Oh, yeah.
he's like, so yeah, when he started, and we were afraid. We had one that broke right over our driveway, so we couldn't get our car out for like days.
Come on in.
Oh.
Come on in. There we go. Yeah, we got a critical mass now. Here we go.
I'm gonna be gone like 2 minutes. I'll be gone.
Welcome.
My
I'm Juliet
professor.
Swaffles. Nice to meet you.
I have like tons of work.
Hi.
You can want it.
Okay. You all may continue eating. I'd like to call the meeting to order, if I may. I'd like to welcome the committee and Dr. Bhattacharya. I hope I said that correctly, or close. Yeah. All right. Dr. Bhattacharya is the graduate dean's representative to the meeting. What I'd like to do is introduce Devalina Goswami in just a second, but this kind of set the rules for the meeting. I'm going to, after the introduction, we'll ask Devalina to do the presentation of the data of the dissertation. And following her presentation, any of the people here who are not members of the committee are welcome to ask questions. Ask very hard questions if you want to, that's fine too. Not just the easy questions, but hard questions. And then after the people who are not members of the committee, I'll ask to have you excuse yourselves, and then the committee, Devalina's committee will quiz her individually, okay, in executive session, okay? So, with your concurrence, we'll begin, okay? So it's my privilege, really, I think it's kind of a privilege to, because I haven't done it in a long time, to introduce a doctoral student to do a dissertation who has sort of worked under my wing a little bit over the last just year or so. Devalina Goswami got her bachelor and master of science degrees from the University of Calcutta. And somehow she worked her way to Lubbock, Texas, and worked with Dr. Shu for a while. And then came over one day and asked if she could come over and do a little selenium work. And I said, okay, come on over, we'll try to get something for you. So Devalina has worked very hard, I think, for the last year. In addition to this research work, she's also been teaching. So she's also had teaching duties too. 1325, right? 1325, which she's taught this last semester. So she's been a very busy individual. So with that, Devalina, I'll turn this over to you, and you can do your presentation. I've asked her to keep this somewhere between 45 and no more than 50 minutes, okay?
Welcome, everybody. Thank you very much for coming here. It's a very bad weather. I did not really pick I promise. This is my title of my dissertation, cytotoxic effect of selenium conjugated transferrin on leukemia cell line. And today when I'm here, I really want to share one thing. Exact 1 year before, exact this time and this day, I defended my proposal. And here I am with my all results and all my data presenting my doctoral defense. So these are my dissertation contains with two types of human leukemia cell line, human transferrin and human serum albumin, and selenium conjugated ester, that's the vital guide here. Let's begin with the leukemia. Leukemia first described in 1985 as immature cell populate bone marrow, lymphoid tissues, and blood. Nowadays, the definition of leukemia is heterogeneous clonal malignancy of hematopoietic stem cells with an abnormal cell survival, impaired differentiation, and with often uncontrollable proliferation. Just for the information, other cell lines, other cancerous cell lines also propagate uncontrollable proliferation shows. This guy over here, this is Sidney Farber, who first tried to treat leukemia children patients with meds, which is known as chemotherapy nowadays. His first med was aminoterine, which is covered in 1947. From his laboratory he developed, but aminoterine, what's the situation, what's the condition of all the leukemia patients? Because the leukemia cells were using the folic acid for their development. Then in 1950, near end of World War II, he and his colleague developed a new medication for leukemia, which is an analog of folic acid, methotrexate. There are different kinds of leukemia we found. Among them, the rarest type of leukemia is chronic myeloid leukemia and acute monocytic leukemia. And acute monocytic leukemia also can be called like acute myeloid type of leukemia. They both are associated with chromosomal translocation or chromosomal abnormalities. Chronic myeloid leukemia, or CML, is associated with 9 and 2 chromosome translocation, and AML, or acute monocytic leukemia, is associated with chromosome 11 abnormalities. This is a small graph that I found in web that's showing leukemia rates in children and young adults in United States from 2005 to 2009. I'm just not going to go over so much. This is the red line showing the CML rate, and the AML rate is right here. This is just a short slide about the normal chromosome, how they look like, and the CML chromosome, and also when the 9 and 22 chromosome get together or translocate, that is called Philadelphia chromosome. My main work with in vitro studies, so I work with all the 2 types of leukemia cell lines. They are isolated. Human chronic myeloid leukemia cell line were isolated from a 53 years old female patient, and then they are characterized and studied a lot. And then the THP1, or acute monocytic leukemia, that is isolated from 1-year-old boy, and then their characteristics are explored. These are the 2 cell lines that I used in my research throughout my whole research. There are certain reasons to choose these 2 type of cell line. Depending on this article, they published on 2007 by Calzolari et al. They showed that different kinds of leukemia cell line overexpress with TFR, transferrin receptor 1 and transferrin receptor 2. So leukemia cell lines are a little bit different from the normal cell line because of their cell signal transaction and uncontrollable cell proliferation. And studies have shown that the 2 cell lines that I have used, it's really overexpressed with TFR or transferrin receptor. Due to increase their endogenous iron requirement, the cancerous cell line used to found with overexpression with transferrin receptor. So what is transferrin? We all know transferrin is an iron transporter, and also it helps to prevent or protect our body from the iron toxicity. If the iron present freely in our body, that could happen a toxicity. So chelate form or chylate form of transferrin, it always helps or protects our body from any kind of toxicity. Transferrin is a single chain polypeptide with approximately 700 amino acids in it, organized with C and N lobes. That is a glycoprotein. Approximately the weight of the protein is 18 kilodalton, and it consists approximately 58 lysine residue in it. Why the lysine residue is important, we will talk about this thing later. This is a short picture of the iron transporter system. Ubiquitously transferrin receptor are spread through the cell membrane, and holotransferrin, which is a form of iron-bound transferrin, is recognized by the transferrin receptor and in the cytosol inside of the cell. And due to the change of the pH, iron gets released inside of the cell, and then it transported back the transferrin receptor into the cell membrane to accept another set of iron-bound or holotransferrin. So this is the main transporter system, and on the basis of this system or this mechanism, we build up our part of the research. Selenium, the most important guy.
Are we done now?
So selenium is famous for its toxicity for a decade. This guy over there is Marco Polo, who first observed selenium toxicity in horses while he was traveling through China. And then later on, a lot of studies takes place, and then Dr. Schwartz, in late 1960s, he discovered first time that selenium has its nutritional benefit in our system, and he tried it on rats. He first found out this nutritional benefit of selenium in rats. So dietary requirements for any adults is 55 microgram per day, and the toxicity level is if we exceed 5 milligram over 5 milligram per day, that could show the toxicity level. The first discovered selenoamino acid is selenocysteine, which is mostly found in bacterial foods, and also selenomethionine, mostly found in plant and animal foods. This is a crystal structure of glutathione peroxidase. Why I'm saying that, I will just talk about in the next slide. Glutathione peroxidase is a very important antioxidant enzyme in our body, which helps to protect our body from any kind of oxidative stress. What it does, glutathione peroxidase helps to convert H2O2 to form H2O in presence of, and use glutathione as its cofactor, and decrease the oxidative stress. That's the main mechanism of glutathione peroxidase here. Next slide. This is a very important slide for our research. In 1988, Sewell et al. First discovered that selenium can produce selenium can be reactive, and selenium can produce some oxidative stress and superoxide. And later on, in 1992, Chaudier et al. Detailed all the relationship of the toxicity of selenium and production of its superoxide in presence of glutathione. This is the redox cycling that he showed. Here, the selenide right here, which reacts with glutathione and produce a superoxide in the biological system. If a selenium compound cannot produce selenide, they cannot produce superoxide. And we depending on this redox cycling, we develop our research, and the selenoester that we are conjugating to our transferrin, that selenoester capable to produce superoxide in presence of glutathione. So, my hypothesis of my dissertation is that covalently conjugated selenium, epotransferrin, holotransferrin, and human serum albumin will decrease cell viability in human leukemia, K562, and THP1 cell lines. Let's move on. The methodology that I followed, the first important thing is selenium conjugation. In 1970, first we discovered Bolton-Hunter reagent. I know many people are not familiar with this name. It's very old assay for radioimmune assay when we do not have ELISA kits. We modified our Bolton-Hunter reagent with the selenoester, with this selenoester Selenocyanate ester, the propionic acid. It's a big long name. I'm just gonna go by selenoester. This guy go and attach to the lysine of the transferrin, and that is why the amount of lysine in the transferrin, it's very important, because this guy is attacking those lysine and making a covalent conjugation over there to make the drug or the treatment. So, cell culture, I am very famous with cell culture. Everybody can see me on my page. The K5 and the 62 and THP1 cell lines were maintained in 10% supplementation of fetal bovine serum with RPMI 1640. These are the just normal with phenol RPMI 1640. And then we suspend that in suspension of 5% of CO2 at 37 degrees Celsius, and we make sure 95% of humidity is there. Cell density, I always maintain my cell 1 × 10⁻⁶ cells per ml in a flask, and if it is exceeding that, I passage those cells. The fresh growth medium always uniformly change